Okay, so today we are finishing up our unit on protists. Remember, we are in the eukaryotic cells in the domain eukarya, so that's where we're headed for the rest of today. If you will take out your chart where you had the supergroups, we're going to continue with that today. So in our last lecture, we covered the archaeoplastids. Remember, we talked about our red and green algae. Those were the protists and the archaeoplastids. And then in the chromoviolatas, we talked about the brown algae. Today, we're going to be talking a little bit more about chromoviolata. And specifically within chromoviolata, we're going to be looking at these two subcategories. We're going to start with the stramenopiles, and then we're going to move on to the alveolates. But stramenopiles, if you remember, that word stramenophile means like hair strewn into a pile, like for bedding, either hair or straw. So that's what that means. But we are into the diatoms today. Just as a reminder, we are still right now looking at the plant-like protists, the photoautotrophs. So that's where we're continuing with our diatoms. Okay, so our diatoms, like we said, they're in the group stramenopiles. Diatoms are single-celled organisms, but they have a cell wall that's made out of silica or glass, and they are very ornate looking. They are a part of the plankton, which means that top layer of the ocean or even of freshwater that are the photoautotrophic levels, so they have lots of light in that top layer of water. They are hugely numerous. They produce a large quantity of oxygen. In fact, they actually produce 20% of the Earth's oxygen. They produce sexually or asexually. So this is just an example of what some of them look like. Looking at them up close, one thing that separates diatoms from some of the other phytoplankton is that they have an upper and lower half of their cell walls. So each one, as you can see, has an upper and lower half. They fit together. And then they have a bunch of pores of various shapes and sizes, and that's how the organism interacts with its environment, is through those pores. And so that's kind of how you can identify a diatom. If you were looking under the microscope, you would definitely want to look for the two halves. And I just included this in here just to show how very ornate they are in the varied shapes that they come in. And so because of that silica, they have just a gorgeous look to them. Sometimes they have like a rainbow effect when you look at them. But they are essential to oxygen on Earth. And you're going to definitely see that later. I'm going to have you watch as an additional assignment a particular video by National Geographic. And it starts with this very thing, that a lot of our oxygen is produced by diatoms. Also important to know sort of their ecological role, not only as oxygen producers, but also as the phytoplankton base of fresh and marine water food chains. From an economical perspective, diatoms have a huge commercial use in what we call diatomaceous earth. It is a bug repellent because it's abrasive, so the diatoms act as an insect killer by pulling oils out of an insect's exoskeleton, which dries them out. And the abrasiveness of the silicone cell walls, because they're made of silica, speeds up that process. So it essentially kills the insects by drying them out. It kind of acts as a desiccant. So that's diatomaceous earth. Okay, so that was what I wanted to touch on with the stramenopiles. We went through brown algae and diatoms. We're not going to go through water molds. We're going to move on to alveolates, and this includes our dinoflagellates, our ciliates, we're going to focus on the dinoflagellates. So again, we are still photoautotrophic. So dinoflagellates are photoautotrophic algae, and they're encased in cellulose and silica. So unlike diatoms that are just in silica plates, dinoflagellates also have cellulose. So this is what they look like, and grab my little highlighter pen here. So dinoflagellates are unique in that they have 2 grooves and 2 flagella. And the way the flagella work is, let's see right here. We have one flagella right here that spins and moves the organism forward, and then the other flagella acts as a rudder to steer them. So this unique structure makes them called dinoflagellates, and here are the 2 grooves, this direction and then of course this direction, and that's for the flagella to be in. Now the reason they're called alveolates is because they have what's called alveoli, which are air sacs that are just beneath the plasma membrane, and that allows these organisms to float in the water column because they are again a part of the phytoplankton. They are mostly in marine environments, but some are in freshwater as well. But those alveoli help them to float towards the top of the water column, allowing them to be closer to more sunlight because they are photoautotrophic. This is just kind of one cool thing. They can be bioluminescent. I have yet to be able to paddle through something like this, but I would love to. So if you have, I think that's really cool, and I'd love to hear about it. But here is another problem. We have 3 dangerous genera of dinoflagellates. And one of your additional assignments is this week's additional assignment about Pfiesteria. And that's one of them, the Pfiesteria species. It produces toxins that cause lesions in humans and in fish. So Pfiesteria was responsible for a lot of fish kills in the mid-80s to mid-90s. They secrete like a neurotoxin that kills the fish when it's present in large numbers. Once the fish are dead, the organisms then act as saprotropes. So they kill the fish using their neurotoxin, and then they ingest it. Once they finish sexual reproduction by forming cysts that fall out of the Pfiesteria and into the water column. So they're dangerous to fish and humans. Their populations tend to bloom when there is a lot of extra nutrients in the water, particularly phosphorus and nitrogen. So when you read the article for your additional assignment this week, just notice that this was a problem in the Neuse River, and that's why I chose that article, because it was local, and I actually know the author. Joanne Burkholder was very popular when I was in college. She did a lot of work with many of my professors, so I heard about Pfiesteria research all the time, and so I would just like you to get to know it, mostly because it was a local problem that was caused by a lot of pollution, but it's been cleaned up, but we need to always know about it so we don't repeat history, right? Okay, this is another one of the dangerous genera of dinoflagellates. And it causes it's called paralytic shellfish poisoning, which is a respiratory failure. So saxitoxin is a poison that's found in certain edible mollusks at certain times. And so that is produced by the I can never say this word correctly, Goniolax species, and it's consumed by the mollusks and the fish. and they don't have any ill effects. But it's a neurotoxin, and it causes respiratory paralysis in mammals, and so therefore it is known as shellfish poisoning, because those who eat the shellfish then have respiratory paralysis and die. Okay, the third you may have seen before. Gymnodinium causes what we call the red tide, which causes fish kills. And if you take a look down here at the bottom, if you see that dinoflagellate, you might notice something a little different. It doesn't have those silica plates, so it's called an unarmored dinoflagellate. And it's a marine species, and it does produce toxins that we associate with the red tides. Usually in the Gulf of Mexico or off the coast, they're off the coast of western Florida. And so far, no deaths have really been associated with that, just respiratory irritation, because that toxin gets aerosolized when the waves are crashing and that sort of thing. So it irritates people's respiratory system, but so far no deaths have been caused by that. Okay, so now we're going to move into the supergroup Excavata that mostly contains zooflagellates, and they have often have a distinctive oral groove that kind of distinguishes them to be in this particular supergroup. And one that we use often to represent this is the euglenids, or a common name Euglena. So they have two modes of nutrition. So far we've been talking about photoautotrophic protists. When we get to Euglena, we have two different options. They are sometimes photoautotrophic, but only 1/3 of Euglena actually have chloroplasts. But they can also be heterotrophs, so they can cross over between the two. Some unique features about our Euglena is they have either atypical mitochondria or no mitochondria at all sometimes. And then they have what's called a pyrenoid, where carbohydrates are stored as paramylons. So taking a look at this picture over here to the right, some features that you're going to see are the eye spot here. That eye spot is a photoreceptor that allows the Euglena to move towards the sunlight, because if it has the chloroplasts, it wants to be able to get closer to sunlight. Otherwise, it can sense where its food is and move towards that as well. And so while we're talking about movement, it has this long flagella in order to move. It has what's called a contractile vacuole, and that is right here. The contractile vacuole actually helps to regulate the amount of water within the Euglena because it is a single-celled organism. So that's what that is for. It does have a nucleus, of course, because it is a eukaryote. And then the pyrenoid that we talked about, where it synthesizes those paramylons and where it stores its carbohydrates. So those are some of the features of our Euglena. When we get in lab, you're going to have a worksheet very similar to this to make sure that you know kind of the ins and outs of the Euglena, the anatomy of it, and what it looks like. And then we will also be looking at live specimens in the lab. So if you will flip over to the link for this video, or you can possibly press play. I'm not really sure if that'll show up on your video. This is just for you to get an idea what the Euglena look like, because you haven't seen it in lab just yet, so this will give you a chance to see it. Okay, so far, all of the protists we've been talking about make up what we call the phytoplankton. And they have been photoautotrophic, or with the Euglena it does cross over a little bit. But just so you know, phytoplankton is all the photosynthesizing organisms in the ocean's, at the base of the ocean's food web. So we have plant-like protists, like we've been talking about, and we have cyanobacteria. Those two things together make up the phytoplankton and are responsible for 50% of all the Earth's oxygen production. So even though they're microscopic and you can't see them with the naked eye, they are so important to our existence on Earth. Now we're going to move on to our animal-like protists, those that are heterotrophs. Remember, Euglena kind of crossed the line between photoautotrophs and heterotrophs. So our animal-like protists are called Protozoa. So our protozoans, like we said, are heterotrophic. They ingest their food. And here are some characteristics of our protozoans. Most members of this class are motile. And often that is how we classify them, based on their mode of motility. They can reproduce asexually by fission or sexually, but usually that only happens when they're under stress. Most of them are unicellular. There are parasitic species that can form resistant structures called cysts so that they don't get wiped out. And then many of them have the contractile vacuoles, like you saw in the Euglena, that regulate the water within their cells. So here are the 4 types of motility that are seen within our protozoa, and like I said, this is how we classify them. Movement by flagella, by pseudopods, and by cilia. Remember when we were talking about prokaryotes, we said cilia are only found in eukaryotes, right? So this is where they show up in the protozoa. Okay, I know you keep seeing this picture over and over again, but right now we're going to stay in the supergroup Excavata as we move into the protozoa. So movement by flagella. Species in this group are really known for causing human diseases. So these are the bad guys. Our example of that is Trypanosoma species. It causes African sleeping sickness. There's also Chagas, which is a disease in Central or South America. But we're focusing today on Trypanosoma. And I'm going to give you just a quick, easy way to remember this. If you have a big American Thanksgiving dinner and you have turkey, people always say that turkey makes you sleepy because, do you remember? Tryptophan. So I associate this with African sleeping sickness is Trypanosoma. So hopefully that'll help you remember that. So this is what it looks like, and you will be seeing this one in lab as well. And the way it works is that it cuts off the blood supply to the brain, and that's what makes you really sleepy. In severe cases, people can die of it. Both Trypanosoma and Chagas disease require a vector, which means an intermediate host. With African sleeping sickness, the vector is a tsetse fly, and that's how it's transmitted. For Chagas disease, which causes swelling and heart disease, the vector is a tritomine insect, so it looks a little bit like our stink bugs. And these are diseases mainly in Central and South America. This is often why many diseases that are caused by these protozoa, we try killing the vectors so that they can't be transmitted. Giardia. If you are a hiker, and I am, you know all about Giardia. It causes infections in the intestines that have abdominal cramping, bloating, nausea, lots of diarrhea. Not something you want when you're like trying to hike the Appalachian Trail. This parasite is found worldwide, especially in areas with poor sanitation. So this is why if you are going hiking, particularly on the Appalachian Trail, and you need clean water, you need to bring something to clean the water with, whether that be iodide tablets, a SteriPen, specific filters. It's to get these protozoas out of the water so that you don't get sick. There we go. SteriPen on the left, iodine tablets on the right. Okay, another one is Trichomonas, and this is an STD in developing countries. Lots of people know it as trick. All right, our second mode of motility. Note that all the ones prior to had flagella. Our second mode of motility is going to be pseudopods. Pseudopod means false feet. Okay, so pseudopods are usually used for motility, but they're also used for feeding through phagocytosis. And these species are usually aquatic species. And there are three common names that they go by, and they're used to describe species with pseudopods. Some of them may sound familiar to you. These are the Amoebas, radiolarians, and the foraminiferans. Again, we're going to see these in lab. Just to show you where these fit in our supergroups, the amoebas, of course, are going to be in the Amoebazoas, and then down in supergroup number 6, the Rhizaria, that's where the radiolarians and the foraminiferans are. So we're going to go through those, and we'll hold off on the Apisthoconta until next time. So here is our phagocytosis. Just to kind of give you an image of that, probably like you to take a minute just to look at it and make sure you understand how that works. Should be a review for you. So here is an amoeba, and you can see their pseudopods sticking out. Let me just get my pen so I can show you that. Here we go, right there. Those are the pseudopods, how they move. And again, you'll be seeing these in lab as well. But there's a large diversity among the amoeba. The one on the top left is something that most people see. That's the most common. But as you look at this, there's a wide variety of body types. So the amoeboids have that thick pseudopod that we were just looking at. And then some of the other things that should look familiar, your contractile vacuole, which helps take care of water regulation. Our mitochondria, because we are in a eukaryotic cell. Here's some of those pseudopods. Still has a nucleus and a nucleolus, and it has food vacuoles, where some of that digestion takes place. So here are just some representatives of different amoeba types, and we're going to take a look particularly over here at the Nagliaria. Okay, Nagliaria. This is the life cycle of a Nagliaria. We start in the flagellated body form here, and as it matures, here we go, it's amoeba shaped. Now, it can then reproduce by creating cysts, but but it can cause infections of humans going through the nasal passage and up into the brain where it feeds. This is going to sound really familiar to you because do you remember the brain-eating amoeba that they talked about at the Whitewater Center? It was all over the news because a poor teen from Ohio did die from this amoeba, and it was in Agliaria. So we need to talk about a few things. That protozoa is in pretty much every water environment around. It does not normally go into your brain and eat you. If that is going to happen, it has to be forcefully shoved through your nasal passages up to your brain. And places like the Whitewater Center do have a lot of force. Typically, that can happen with people who have a compromised immune system already. So that actually was the case for this teen. And it was also a particularly high concentration at the Whitewater Center at the time. The water used to come out of the Watauga River and be circulated through and then taken back out. But the Whitewater Center is unique in that it has concrete walls and it gets very warm. So that's a perfect breeding ground for these type of amoeba. So it's not. So they went through all kinds of precautions. Now, when you paddle down there, or you don't really swim. When you paddle down there, you will smell it. It's chlorinated. It's just like a pool now. Instead, it's not river water that gets circulated in and back out. So it is treated particularly for the protozoa. But here are some other things. If you ever see a whitewater paddler, of which I am one, you will note that they tend to wear nose clips. And as silly as they may look, they accomplish two purposes. One, it does keep those types of things from going up into your nose. No amoebas, no bacteria, that kind of thing. It also, because when you're in whitewater, you do tend to go upside down and have to roll back up. It keeps the water out of your nasal passages so you don't feel like you're drowning. You're not going to drown usually that way anyway, but people like to keep water out of their sinus cavities. Okay, thank you for letting me talk a little bit about whitewater paddling. It's always exciting to me. Moving on into the radiolarians. We're passing the amoebas. Radiolarians. Radiolarians have very thin pseudopods. They have an internal skeleton that's made up of silica. Silica should sound familiar to you, those glass-like products. And so the pseudopods come out through pores, and that's how, again, the radiolarians interact with their environment. So their internal skeletons are called tests. And so the tests of a radiolarian is made up of silica. So contrasting that to the foraminiferans, they also have an internal skeleton that's called a test. They also have very thin pseudopods that come out through pores, but their skeleton is made up of calcium carbonate. So their test is calcium carbonate. Radiolarian's test is silica. That's the difference between these two groups. Again, they're very ornate, similar to diatoms in that sense. So in the lab, you want to be able to tell the difference between radiolarians and foraminiferans versus diatoms. Remember, diatoms have those two halves to their tests or their shells. Okay? So this is something just interesting for you to know. The White Cliffs of Dover in England, that is from millions of years worth of foraminiferan skeletons layering up, because remember, they're out of calcium carbonate, so they make this beautiful white surface. Okay, moving on to movement by cilia, aka the ciliates. They can reproduce asexually by binary fission or sexually through conjugation. And some can even shoot poison out to catch their prey. We will be looking in the lab at two. One of them is called Stentor and the other is Vorticella. So we will take a close look at those. In this picture here you can see two of them. Here's our Stentor and here is our Vorticella. And like I said, we'll be looking at those in lab, but they move via cilia. So just to kind of show you where those are, the ciliates are alveolates in the supergroup Chromoviolata. So they're in both of those two. In Chromoviolata. So they're included with the phototrophic protists because they are also phototrophic. Am I confusing you enough yet? This is why I had you take out that chart so you can write all these things down. Okay, so our best known example of a ciliate is our Paramecium, and I'm sure many of you have seen these before, but we will be looking at those in lab as well. Okay, so here is our anatomy of the Paramecium. And so when we're looking at it, we have the gullet or the oral groove. That is where it ingests its food. And then, of course, the cilia for movement. It has at least two contractile vacuoles, a posterior one and an anterior one. Those are always recognizable because they look very star-shaped, and again, those are still for water regulation in the cell. And then what's unique to our Paramecium is they have a macronucleus and a micronucleus. The macronucleus is the center of all metabolic activity. The micronucleus is it's the storage site for the germline genetic material. So it can give rise actually to the macronucleus as well and is responsible for genetic reorganization that happens during conjugation. So both types of nuclei contain the full complement of genes, so they have all the hereditary information that they need. But the important way to remember it is that the organism cannot survive without the macronucleus, because that's where all the metabolic activity is coded for, but it cannot reproduce without the micronucleus. So you want to make sure you can not only distinguish between the two, not necessarily under the microscope, if you can, that's awesome, but to know what the role of each one is. Okay, our protozoa that basically have no movement. They have no way of moving on their own. And these are what's called the sporozoans, which should kind of give an idea as to how they get around. They spend a large portion of their life cycle as spores, or cysts, one or the other. And their life cycle usually has multiple hosts. So the first one that we want to look at is toxoplasma. Let me back up and say all of the protozoas that fall into this category are parasites, and they contain what's called an apicoplast, and that's used to penetrate their host cell wall. That's how they infect their host. So toxoplasma, or it causes what's called toxoplasmosis. Its hosts are cats, pigs, rats, and humans. So this is the life cycle of it. And if you have ever been pregnant, you have probably been to your obstetrician and they've asked you first thing, do you have a cat in the house? And this is the reason that they're asking that. Because if we look at the life cycle, let me get my pen up again. Let's start with our oocyte. Our oocyst comes from our cat feces. That's, because it was in the host of the cat, It moves through its fecal matter. And then, of course, if you have an indoor cat or an indoor-outdoor cat, you are probably cleaning the litter box. So if you do, then that can contaminate you as an adult. Now, typically speaking, it can make adult humans sick, but it doesn't usually kill them. However, when a fetus is infected through the placental wall, the fetus can die. And that is why your obstetrician will immediately ask you, do you have an indoor cat? If you do, you are not allowed to clean the litter box. So that is our sporozoan. Another sporozoan is malaria, and that's caused by Plasmodium, which is a particular protozoa. And again, we have the hosts, and the host, as most of you will know, is mosquito and human. So what happens is, if you can look in this picture pretty quickly, let me get my pen again. You can see how the zygote is here in the gut of the mosquito. All of this takes place, and then spores are produced. As it is injected into humans, then there's another life cycle there, and it makes people obviously very sick. 300 to 500 million cases per year. 1 million people die each year from malaria. It's why people work so hard to kill mosquitoes, and particularly in areas that are infected with the Plasmodium sporozoa. When I was a kid, I grew up in Fort Lauderdale, Florida, and when I was in Fort Lauderdale, this doesn't happen anymore, just so you know, to let you know how old I am, they used to come spray for mosquitoes using the biplanes. So they would come, like, drop a piece of paper to let you know that was going to happen. So you would potentially go inside, and they'd come spray DDT to kill all the mosquitoes. So it was really a great thing. We didn't have that many mosquitoes. Anyway, so we'll move on. I mentioned that almost all of these sporozoas have what's called the apicoplast. And so if you look in here, they have, let me find my pen again. It basically is this needle, hypodermic needle, that shoots out and injects its host. And so that is how they infect their host. So the apicoplast has become a target for anti-malarial drugs. If you can kill that, then it doesn't matter how many times you get bitten by the mosquito, you are unable to be infected. So that's where a lot of your anti-malarial drugs come from. Okay, so we just went over our animal-like protists, and there are 4 different types of motility. Lastly, for our protists, we're going to talk about the fungal-like protists, or the saprophytic. These are our decomposers, or the absorptive heterotrophs. And there are 2 different types. Just so you know real quickly before I give you those 2 types, they fall into the Amoebazoa supergroup. Okay, so the first one is plasmodial slime molds. Their scientific name is Physarum. Now, do not confuse this with the species name or the scientific name Plasmodia, which we already talked about causes malaria. That is different from the common name Plasmodial slime mold that we see here that goes with the scientific name Physarum. So this is an amoeba-like single-cell organism, and it engulfs bacteria and other food, and then it can dry up and form spores for reproduction. And we're going to see this in lab as well, so let you know that. On the next slide, there's a link to a video just so you can kind of see what it looks like. It's a time-lapse video of it engulfing its food and moving out. Okay, the second type is a cellular slime mold. And these are individual amoeba-like cells. And they live in the soil and they eat bacteria and yeast. And they may look familiar to you because oftentimes if you put down mulch, they tend to like mulch, and usually there are spores of them within the mulch, and so once it's wet, then they can begin to grow and eat the bacteria and yeast on the mulch. So this is what it looks like when it's dried and starting to produce spores. A lot of times it's called dog vomit fungus. That's always a pleasant name. But that's what it looks like. If you've seen that kind of yellowish dried up stuff on your mulch, that's what you have. So that completes our unit on protists.